Am I audible?
Yes, ma'am, you're audible.
Okay, so I don't know if anybody else is joining, but I think I will just start the class. Okay, I hope my screen is visible. So, I can start. Okay, so this is week 7, live session for experimental biotechnology from NPTEL. And this is by Professor Vishal Trivedi from IIT Guwahati. I'm one of your TAs, Sumati Ariraj from University of Hyderabad. So this week, whatever the questions were given in the assignment, we will be covering the topics related to that. So the first topic was regarding the antibody production. So antibody production occurs in like different phases depending on how much it has been stimulated, how much antigen exposure has been there. So the different phases can be named as either lag phase, log phase, plateau phase, and decline phase, or it is also known as induction phase, exponential rise phase, steady state phase, and downward phase. So in the lag phase, it is when within the few days of entry of an antigen into the body. So what happens at this time is the antigen is being recognized and then it will be processed and then presented by the APC cells, that is the antigen presenting cells. Then once this is done, activation of B cells and T helper cells takes place. Then because all these process is going on right now, even though the antigen has entered the body, at this phase there will be no detectable antibody in the circulation. So even if there is very minute quantity that has started to be produced, but it won't be detectable, it is that less. But after this, when the B cells are activated properly and they proliferate and the plasma cells start secreting the antibodies, then we have exponential rise in the antibody phase. So here, this phase is after the lag phase, after the recognition process by APCs and then activation of B and T cells. So the activated B cells will proliferate and then differentiate into plasma cells. Now the plasma cells will secrete large amount of antibodies. Then, because of this huge production in the antibody titer in the blood will rise steadily and exponentially. This is the phase where the immune system mounts an effective defense because your antibody titer is highest in this phase. After that comes the plateau phase. So here what happens, the antibody production continues but reaches a balance with utilization and production. So once produced, the antibodies has to be then bound to the antigen. It has to continue the immune pathway. So few antibodies will be in the process with the antigen, and few are continuously being produced, which are now yet to bind to the antigen. So both the newly produced antibody and the antibody which are being utilized or which are undergoing degradation also, all are happening simultaneously. So it will be in a balanced form. How much is being depleted, the same amount will be produced. So it will be in a steady state, that is in balanced state. Then antibody titer remains relatively constant at the peak level. So once the peak is reached, then it will go to a plateau and it will be constant now. Then comes the decline phase. So once the antigen is eliminated or neutralized, the plasma cells reduce in number and the antibody production decreases. Then the antibody titer gradually falls due to the natural degradation or natural catabolic process. Then, just a second. Okay, sorry. So then, after the antibody concentration or titer gradually falls down, it will go into the downward phase. So you can see here, this is the lag phase, this is the log or exponential phase, and then you have a plateau phase where it will be relatively more or less same, and then there is a decline phase. So this is the initial exposure phase, this is the antibody production phase, and this is where both production and depletion are in a balance. And then once the antigen is being eliminated from the system, the antibodies also start to decline. So the steady rise in the antibody titer happens during the log phase because that's when the plasma cells are actively producing the antibodies after successful activation. So this was about the antibody titer. Then comes monoclonal antibody. So monoclonal antibodies are a uniform collection of identical antibodies secreted by a single B cell. So that is why they are monoclonal in nature, and they are all from one specific antigen determinant epitope. So an antigen might have more than one epitope or multiple epitopes on its surface, and different B cells can recognize different epitopes and produce antibodies specific to that particular epitope. So generally, in a population of B cells, you will have different types of B cells which have recognized different epitopes and have produced uh, different type of antibody for the same antigen. So this becomes polyclonal because they are coming from different clones of they are clones of different B cells which have recognized different epitopes. But then when you take up one single B cell which has recognized one particular epitope, and then you clone the B cell, then you will get antibodies which are monoclonal now, specific to that single epitope. So, in contrast, polyclonal antibodies are produced by different B cell clones, so they recognize multiple epitopes on the same antigen. So, why B cells? So, only B cells have the ability to differentiate into plasma cells and secrete antibodies. T cells, macrophages, NK cells do not secrete antibodies. So once a single B cell is activated against an antigen, all its daughter clones produce the same antibody. So as I said, because this one single B cell has identified one epitope, if you make a clone of it, it will be exactly same as this cell. So all the clones will also be producing the same antibody to that particular epitope. So this technology or this method to produce monoclonal antibodies is known as hybridoma technology. It was developed by Kohler and Milston, and this method allows unlimited production of specific antibodies. So this single monoclonal antibody will be highly specific to that particular epitope. So there are different steps involved. This is a brief of all the steps. So first is you have to immunize a animal or a model. So, for example, if you have a mice model, you will have a mouse which is being immunized by the target antigen. So, the target antigen will be injected into the mouse so that the mouse immune system will recognize the antigen and produce uh, antibodies against that antigen. So the animal's B cells will now, especially the spleen, that will produce antibodies against the antigen. Then we have a process known as cell fusion. So spleen B cells are short-lived, so they have to be fused with cancer cells like myeloma cells. So particularly myeloma cells are used. These are immortal cancer cells which do not produce any antibodies. So these are only used for their purpose of uh, making the B cells become immortal because they are short-lived and will die, and you cannot use it for antibody production unless it is fused with a myeloma cell. So now fusion is carried out using PEG, which is polyethylene glycol, and then hybridoma cells form. So you can see here you have your spleen cells. So these are different colored because to represent the different epitope that each cell has identified, and each cell will produce a different type of antibody for the same antigen. So from here a single cell will be picked up, and then it will be uh, fused with the myeloma cells, and then you get hybridoma cells. So the fused cells combine will now have properties of both B cells and myeloma cells. So from B cells, it has the ability to secrete specific antibodies, and from myeloma cells, it has gained immortality. So it can be continuously grown in the culture to continuously produce the antibody. Then we have to select the cells. So now after fusion, you will have some cells which have not fused. So you'll have spleen cells which have not fused. You'll have myeloma cells which are not fused, and then you'll have a hybridoma cells which is fused. So now you have to select particular cells from the mixture of all these. So you need only the hybridoma cell. So how do we do that is we use something called as hat medium. So the mixture is grown in hat medium, which is hypoxanthine, aminopterin, and thymidine medium. So in this medium, only the hybridomas will survive, and the unfused myeloma cells will die. And the unfused B cells, because they are naturally short-lived, they will also die. Then screening and cloning is done, where hybridomas are screened to find the ones with desired antibody. So now you can see you have a mixture of cells which have different epitope. So now if you want monoclonal, particularly you will select one cell from which you will start to clone the cells. So the positive clones are then isolated and cultured. Then large scale production is done. The selected hybridoma clone is now expanded in vitro or in vivo to produce large amounts of monoclonal antibody. So this is the process of hybridoma. Then the applications are, it can be used in diagnosis, it can be used in therapeutics, it can be used in regular research experiments. So in diagnosis, it can be used for pregnancy tests, it can be used for infections as markers, cancer biomarkers. In therapeutics, there are antibody therapies that are going on. So we have anti-cancer drugs like rituximab and trastuzumab, and then autoimmune disease treatment with infliximab for rheumatoid arthritis, and antiviral therapy parivizumab or RSV. And in research, of course, they are used in different techniques to detect proteins, either in Western blot or in immunofluorescence or ELISA, flow cytometry, immunohistochemistry, etc. So these are the applications of monoclonal antibodies. Next is immunogenicity versus antigenicity. So immunogenicity and antigenicity, sometimes people use it alternatively, but both have specific definitions. So immunogenicity is the ability of the substance to induce an immune response. That is, it will activate your B and T cells, and it will start your immune mediated responses. Antigenicity is the ability of a substance to be recognized and bound by the products of the immune system or the immune response products like antibodies or T cell receptors. Then all immunogens are antigens, but not all antigens are immunogens. This is a very common statement. So sometimes an antigen might not be able to invoke an immune response, so it will not be immunogenic in nature. So that time we can combine it with a hapten molecule to make it immunogenic. So what is a hapten? A hapten is a small molecule, usually, a hapten is an antigen which is not immunogen. So it will be combined with a carrier molecule to make it immunogen. So, a hapten is a small molecule which is an antigen, and due to its small molecular weight, it cannot be recognized by the immune system. So, it has to be combined with a carrier molecule. So, it cannot stimulate an immune response by itself, and it can be recognized by antibodies if the immune system has already been primed against it. So even though it is not able to invoke an immune response, if that molecule in some other form had been already introduced into the system and it has been able to produce an immunogenic response, then your system will have a memory of the epitope of the hapten. So second time, it does not require a carrier molecule. Second time, your antibodies, which are already stored in the memory B cells, that can recognize it, and then it can produce. It can, if the antibody is already present in the system, then it can bind to it. But if the antibody is not present in the system, it cannot recognize and produce the antibodies now. So that is what is a hapten. So examples of haptens are penicillin, urushiol, DNP, etc. So all these are also allergens. So that is why sometimes they also cause allergic reactions. So here comes, like I said, the hapten carrier concept. So a hapten itself is not immunogenic but is antigenic. So when a hapten is combined with a carrier, it now becomes immunogenic in nature. So, hapten alone is too small and simple, and it is non immunogenic. When it is bound to a carrier protein like BSA, KLH, or ovalbumin, these are larger molecules. So, when attached to it, the size of the combined size of the hapten carrier is more, so it can be easily recognized by the immune system and it can activate the immune response. So this is now large, complex, and provides the T cell epitopes. Then haptocarrier conjugate, immunogenic because the hapten provides the B cell epitope, and now the carrier can provide a T cell help. Then together they allow B cell activation and antibody production. So, why high molecular weight proteins are more immunogenic? So, when they are large, complex, they are structurally diverse. They can provide multiple epitopes for both B cells and T cells. So, it helps the strong activation of both B and T cells. So, examples for these are bacterial toxins, viral proteins, etc., which are highly immunogenic in nature. Then, to summarize, antigen is usually immunogenic if it is large enough. If it is smaller than, say, 1000 Daltons, then it won't be immunogenic even though it is an antigen. Then, high molecular weight proteins are strongly immunogenic. Hapten is not immunogenic by itself, but when combined with a carrier, it is immunogenic. Therefore, the answer for the question was hapten. So you can see hapten, they're very small, so these cannot easily provide an epitope or make your B and T cells activate your immune response. But when combined with a larger molecule like a carrier, either BSA or ovalbumin, etc., then the epitopes are exposed to the B cells and T cells for proper activation. So that is why this alone is not immunogenic, but when combined with a carrier is immunogenic. So that was about hapten. Then coming to myeloma cells. So we spoke about myeloma cells in the hybridoma technology. So what exactly are myeloma cells? Myeloma cells are cancer cells, so they are cancer of the plasma cells, which are terminally differentiated B cells responsible for secreting antibodies. In myeloma, the plasma cells become neoplastic, proliferating uncontrollably in the bone marrow. So they produce large amounts of single monoclonal antibody or its fragments in the light chains. So why plasma cells are not B cells or T cells? So plasma cells are final effector stage of B cell differentiation. While B cells can still divide and present antigens, plasma cells are highly specialized only for antibody secretion. So myeloma arises when the plasma cells lose control over proliferation due to mutation. So that is why they become immortal and then they are used in hybridoma technology. Then clinical features of multiple myeloma. So multiple myeloma, we have something like you can remember it like a mnemonic a crab. So we have hypercalcemia, we have renal dysfunction, we have anemia, and we have bone lesions. So hypercalcemia, as the name suggests, is excess calcium because the bone is broken down and it releases the calcium, so calcium levels increase. Then there is renal dysfunction, then light chains damage the kidneys, and then there is anemia. Bone marrow infiltration suppresses normal hematopoiesis, etc. Then bone lesions, lytic lesions, cause bone pain. So these are just some diagnostic clues about myeloma, which is not actually necessary for here, but it gives you an idea about the type of cell that is. Then this is a little more information regarding it. So we have the M protein on the serum and then we have Benz-Jones proteins in urine, this etc. So this is related to the cancer. Then coming to its relevance in immunology and hybridoma. So in lab, researchers use special myeloma cell lines for producing monoclonal antibodies. So like we discussed in the hybridoma technology. So these cells are immortal and they do not produce their own antibodies. So like here it is given that it produces, but when we use it for hybridoma technology, they are engineered in a way that they do not produce their own antibodies. Because if you don't do this step, these cells will also produce their own antibodies and it will get mixed up with the antibodies that are being produced by the B cells. So we have to make sure that these cells are engineered so that they do not produce their own antibodies and avoid contamination with unwanted immunoglobulins. So we need them only for their ability to proliferate continuously and because they are immortal. Then they should also be HGPRT deficient. So remember we had the HAT medium for selection. So why so in this, I said that there will be your unfused spleen cells, unfused myeloma cells, and fused hybridoma cells. And only hybridoma cells survive in the HAT medium, but not unfused spleen cells or myeloma cells. That is because your like I said, the unfused spleen cells will die eventually on their own because they are short-lived. But your myeloma cells are HGPRT deficient, so that is the reason they cannot survive on the HAT medium. So what that is exactly is, uh, yeah, so they cannot survive on the HAT medium unless fused with the normal B cell. That is why only when fused with the B cell, your B cell can survive your HAT medium. So your fused hybridoma cells will now be able to survive and you can easily select them. So the B cells, why fuse with the B cells? So the B cells from spleen, which or with the, of the immunized animals, they provide antibody specificity but are short-lived. Myeloma cells provide immortality but do not have antibody secretion because they are engineered to do so. And then hybridoma, that is the fusion product, has both immortal and has the ability to secrete antibodies from the B cells. So whatever is not here is here, and whatever is not here is here. So that is the reason we need these cells, and they survive on the HAT medium. So myeloma is basically cancer of the plasma cells, and this is engineered in such a way that we can use it to produce monoclonal antibodies. So this is the lineage of cells. So these are blood stem cells. Then you have the lymphoid stem cells. Then you have lymphoblast. From lymphoblast you have B lymphocyte, and from B lymphocyte you have the plasma cells. So this is the cell we are talking about. And there are also other types of cells. You can look through it. Then coming to HGPRT. So why here I didn't explain it completely is because this is the next topic. So here I said that these myeloma cells are HGPRT deficient, and that is the reason they cannot survive on the HAT medium. So what is HGPRT? HGPRT is hypoxanthine guanine phosphoribosyl transferase. So it's an enzyme. So the enzyme has a central role in purine salvage pathway. So purines are nothing but nucleotides. So normal function of this enzyme is to convert hypoxanthine into IMP, that is inosine monophosphate, and then converts guanine to GMP and uses PRPP as ribose donor. So why this is important is the cells need nucleotides for DNA replication, and if the de novo pathway is blocked, they can still survive by recycling using HGPRD. So you have two basic methods. So for DNA replication, that is the required nucleotides. So you have your de novo synthesis, and then you have your salvage pathway. So usually we have the de novo synthesis which is used, but if the de novo synthesis is not there, then the cells shift to salvage pathway where they use GMP, IMP, AMP. So in the HAT medium where you have hypoxanthine, aminopterin, and thymidine, it selects only the hybridoma cells because uh, your HGPRT is inactive in your myeloma cells, which makes them unable to go into the salvage pathway. So only when fused with the B cells still have functional HGPRT. So they can go to the salvage pathway and they can still survive in the medium. So that is how we can select specifically only hybridoma cells. So the components of HGPRT are hypoxanthine, which provides purine base for salvage pathway, aminopterin, which blocks the de novo synthesis of purines by inhibiting the dihydrofolate reductase. Then we have thymidine, that is bypasses the thymidylate synthesis block, which is also part of salvage pathway. So basically, first we block the de novo synthesis, and then we have the hypoxanthine and thymidine, which provides purine for salvage pathway, and this bypasses the thymidylate synthesis block. So all these three together help the cells which have active HGPRT enzyme to survive. So the cells which have active HGPRT enzyme are B cells, which when fused with myeloma cells, they can survive into this. Otherwise, unfused myeloma cells which do not have HGPRT cannot switch to salvage pathway, and de novo is blocked in them, so they die. And then unfused B cells, anyways, they are short lived, so they also die. So this is helpful in selection of only the fused hybridoma cells. So the selection principle is unfused myeloma cells. These are HGPRT deficient. We genetically engineered them so that they are deficient of HGPRT and cannot use the salvage pathway. They die in the HAT medium. Unfused B cells have functional HGPRT, so they could survive in the HAT medium, but they are short lived and die naturally after a few days in the culture. But the hybridoma cells, which is B cell plus myeloma cells, myeloma cell provides the immortality, B cells provide HGPRT, so they are both now long lived and can also go to the salvage pathway and produce the purines. So, produce the nucleotides. Then, together they survive and proliferate indefinitely in the HAT medium, and as a result, only hybridoma cells survive. These are screened for production of desired monoclonal activity. So, other than this, HGPRT deficiency also occurs in humans, and it's called as Leach-Nyhan syndrome. It leads to accumulation of uric acid, gout, kidney problems, neurological effects, etc., etc., because it affects the purine metabolism. So this was about that, and this is a just a brief about the de novo and the salvage pathway. So cells need nucleotides, basically both purines and pyrimidines for DNA RNA synthesis. So they can obtain them either by de novo pathway or salvage pathway. So de novo is from scratch. So the synthesis starts from simple precursors like amino acids, ribose 5-phosphate, carbon dioxide amines, etc., and it builds purines or pyrimidines step by step. And it's energy expensive, and it also requires tetrahydrofolate as a cofactor. Then we also have salvage pathway, which recycles to produce the nucleotides. So it reuses free purine pyrimidine bases that are already present or nucleosides from degraded nucleic acid. So the key enzyme for salvage pathway are HGPRT, APRT, and thymidine kinase. So HGPRT salvages hypoxanthine and guanine, and APRT salvages adenine, thymidine kinase salvages thymidine. And this is energy efficient when compared to de novo synthesis, because de novo is from base, that is from scratch, and this is a recycling process which will definitely be more energy efficient than the de novo. So now coming to hat medium. What is hat medium? Like we discussed, hat medium full form is hypoxanthine, aminopterin, and thymidine. So aminopterin is a blocker. It's a folic acid analog. So it blocks dihydrofolate reductase enzyme. So without dihydrofolate, there will be no THF, and then there will be no de novo synthesis of nucleotides. Then we have hypoxanthine. It provides purine base for the salvage pathway. So salvage pathway, when it's active, it needs something from where it can recycle it. So this provides that purine base, and it requires a GPRT enzyme to convert IMP to AMP or G. Then thymidine. Thymidine provides the pyrimidine base for salvage pathway. It requires thymidine kinase to convert TMP to TTP. Then uh, this we have already discussed, that myeloma cells cannot use salvage pathway. De novo is blocked and they die. The normal B cells can use salvage pathway but are short-lived, but also die after a few days. Then hybridomas only survive because they have inherited immortality from myeloma and HPRT from B cells. So they can use the salvage pathway even when de novo is blocked. So they survive and proliferate in the hat medium. So hat medium blocks the de novo pathway basically of nucleic acid synthesis using aminopterin and forces the cell to depend on the salvage pathway. This is why only hybridomas survive, and we can easily pick the hybridoma cells, which we can further use for monoclonal antibody production. So next is peritoneal exudate cells. So PEX cells. So PEX are immune cells which are harvested from the peritoneal activity. Just a second. I'm still connected. Yeah. My voice is breaking. Is it still the same? I'm sorry, I don't know.
Now it's fine.
Now it's fine. Okay. Do you want me to repeat any part where it was not audible? I can do that. Just let me know.
No, ma'am. Nothing.
Okay, okay, great. So the next is the PEG cells. So yeah, so PEGs are immune cells harvested from the peritoneal cavity of the animals, like whatever animal model you have, either mouse or rat or bovine. So they are collected after injecting an irritant called as thioglycolate broth. So this helps in recruiting the macrophages, lymphocytes, neutrophils, etc. into the peritoneum, and then when you can collect the cells from there. So in hybridoma technology, the pecs from the bovine spleen are generally collected. And why do we need these cells? So these cells are basically feeder cells for the hybridoma cells. So after fusion, newly formed hybridoma cells are very fragile. So we use something called as PEG to fuse the cells, like I mentioned before, polyethylene glycol. So this makes the cells very fragile. So the survival rate of these cells becomes low, and it is very stressful for the cell after the fusion process. So hybridomas cannot initially grow efficiently in vitro. So the feeder cells help provide the microenvironment required for the growth. So they basically help the cells. They give a supporting environment, whatever requirements are there for the hybridoma cells. They provide those requirements, whether nutrients or growth factors or even adhesion. Like cells need adhesion or attachment for survival and growth. So these feeder cells help provide all these kind of environment to the fused hybridoma cells because they are currently very sensitive. So, functions of PECs as feeder cells, it helps in secretion of growth factors and cytokines. So, cells in vitro, they need many factors, they need many nutrients and other things for them to survive and grow properly. So, important things like growth factors and cytokines, which helps the cell to grow and communicate and adhere properly, are all released by PEC cells. So PEC cells release IL-6, which is a growth factor for the hybridoma cells, and it also releases interleukins like 1, 7, and it also releases CSF, GM-CSF. Then it also it helps in the proliferation of hybridoma cells. Then it also helps in detoxification of the culture medium. So the feeder cells help mop up the toxic byproducts like free radicals, metabolic waste, etc. Then cell to cell contact is provided. So they provide substrate adherence and survival signals for the delicate hybridoma cells. So when they have like physical contact with the other cells, the cells tend to proliferate better. So they help in the adherence, and then nutritional support. They also release metabolites that improve cell viability and growth. Other feeder cells are also used other than the PECs, mouse thymocytes, which provides T cell factors, mouse splenocytes, which are rich in lymphokines and growth factors, and rat peritoneal macrophages, which support cytokines, etc. But mostly PECs are only used. So, however, PEC are most widely used because they are easy to collect and highly supportive. So, to summarize, PECs are peritoneal exudate cells from the bovine spleen. So, these are feeder cells for the hybridoma culture, provide growth factors, detoxification, survival support for the fragile hybridomas after fusion. Then, without the feeder cells, many hybridomas would die, reducing the monoclonal antibody yield. So, if you need proper healthy hybridoma cells which can produce huge amounts of monoclonal antibody, you have to provide the feeder cells for them for growth and support. Then coming to the blood types, I think blood types you guys can answer. So the O blood group contains what could be the answer? Can anybody answer? Try to answer this. Does it have A antigen, B antigen? Does it have both or no
Option
antigens?
A.
Yeah, correct. So it has no antigens. So that is why it is O blood group. RBCs which have antigen A are blood group A. RBCs which have antigen B are blood group B, and which have both A and B are AB. So this is the blood grouping. So we have our RBCs, and we have group A which has antigen A. So it will produce antibodies against B, and group B which has antigen B, so the host will produce antibodies only against A. Then group AB which has both the antigens, there there will be no antibodies against either A or B in such type of person. Then group O, which has no antigens, no, no A and no B, can produce both the antibodies against A and B when exposed to the antigens. So the antigens in the blood of group A is A, and group B is B, group AB is AB, and group O, it doesn't have any antigens on the RBCs. In turn, it's reverse when it comes to production of antibodies. Since it has antigen A, when it is exposed to antigens A, B, it can produce only antibodies against B because B is foreign to this person, and since the person already has A. In the same way, in case of B blood group, since the person already has antigen B, there will be no antibodies produced against B. But antigen A is foreign to it, so it will produce anti-A antibodies in the plasma. Group AB, which has both the antigens, will not produce any type of antibodies because both are self antigens. And group O, since it has none of the antigens, it will produce antibodies against both A and B. So this is a table which you should remember to easily remember about the antigens and antibodies regarding the blood groups. So next is overview of ELISA. So ELISA is enzyme linked immunosorbent assay. So the principle of it is it uses the reaction between antigen and antibody and then an enzyme substrate reaction to produce a measurable color which can help in quantification of the antibody, or it can help even to detect the presence of the antibody. Then enzyme that is commonly used for this purpose for the enzyme substrate reaction is HRP, that is horseradish peroxidase, or even alkaline phosphatase. So there are different types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So just a brief about all these. Direct ELISA is antigen is coated on the plate and then enzyme linked primary antibody binds directly to the antigen. So you have your antigen and then you have the antibody against the antigen, and the antibody itself is tagged to an enzyme. So when you add a substrate, immediately color change is observed depending on the concentration of primary antibody bound on the antigen. So this is direct because your enzyme is tagged to your primary antibody itself. It uses only one antibody, that is primary antibody with enzyme, and it is quick and it is simple. But however, it can have like low sensitivity since only one antibody is used. Then indirect ELISA. Indirect ELISA is when you coat the plate with the antigen. Then you have a primary antibody which binds to the antigen specifically. Then you have a secondary antibody which binds to your primary antibody. Then your secondary antibody is linked to the enzyme, and when you add a substrate, then there is a color change. That is why it is indirect, because your enzyme is linked to your secondary antibody instead of primary here. So you need two antibodies here. One is primary and then one is a enzyme linked secondary. So it is more sensitive because you have secondary which can easily bind to your primary now, because your primary has bound to your antigen and now your secondary can bind to it and amplify it. So it is more sensitive. Amplification of multiple secondary antibodies occurs, and then cons is, however, you need to do an extra step. Obviously, you need an extra antibody, extra step, and sometimes it can cross react. So the specificity might be a little less than this, but it is more sensitive. So if your antigen concentration is very low, also your indirect ELISA can help detect it. Then competitive ELISA. So competitive ELISA is where you have an antibody that is incubated with the sample antigen, and then this mixture is added to a plate coated with the same antigen again. So you have your antibody with your antigen, then this mixture is added to a plate which is having the same antigen. So basically, it's a competition between now the antigen bound with your antibody and your coated antigen. So if the sample antigen is high, fewer antibodies left to bind to the antigen. So less antigen antibody will be bound on the plate. So enzyme linked detection occurs in inverse signal. So more antigen, less color, less antigen, more color. Because if you have more antigen, all your antibodies will be bound in your incubation mixture itself. So there won't be available antibodies when you add it onto the plate. There won't be enough antibodies to bind to the antigens on the plate. So your color will be less. This indicates the sample has more antigen and vice versa. Then antibodies here used is usually one, only the primary antibody. Sometimes secondary can be used for detection also. Then it is useful for detecting small antigens and haptens, and it is more complex to interpret. So since it is a reverse of your direct or indirect ELISA, you need extra steps to calculate the amount. Then sandwich ELISA. So sandwich ELISA, as the name suggests, you have antigen, antibody, then uh, your secondary. So you have your plate that is coated with the capture antibody specific to your antigen. Then you have your sample antigen, which binds to the antibody on the plate. Now you have detection antibody. So it is added uh, and it binds to a different epitope on the same antigen. So here in indirect ELISA, you first have antigen, then you have primary, then you have secondary. So it is the order of binding is antigen, primary antibody, secondary antibody. But however, here you have first an antibody already bound to the plate. Then you have antigen in the middle. And then you have another antibody bound on the other side of the antigen. So your antigen is basically sandwiched between two antibodies. So the enzyme linked secondary antibody is then added and the substrate is added and then you get a color change. So here you will need three different antibodies. First antibody is capture antibody, which is put on plated on the surface, and then you have your antigen. Then you have second antibody, which is your detection antibody, which binds to a different epitope of the antigen. And then you have a third antibody, which is your enzyme linked secondary antibody, which will bind to your detection antibody. So you have three antibodies here. It is highly specific and it requires antigen recognition by at least two different antibodies. And it is highly sensitive because amplification occurs because of the secondary antibody that we are using. And it is more expensive because it uses three antibodies and it requires well characterized antibody pairs. So you need to have an antigen which can recognize two antibodies or which can be recognized by two different antibodies. If it has a single epitopic region, then you cannot use that kind of antigen for this. So this is about the different types of ELISA. So you can see here a picture. This is your direct ELISA. This is your plate, ELISA plate. So you have your indirect, you have your antigen on the plate. Then you have primary antibody specific to your antigen, which is directly attached to the enzyme. Now when you add the substrate, enzyme substrate reaction occurs and you get the color. So it is simple, it is direct. Then you have indirect. You have your antigen on the plate. Then you have primary antibody specific to the antigen. Then you have secondary antibody which attaches to your primary. And your secondary is attached to an enzyme. And then when you add a substrate, it will react and give color. So it is indirect because secondary is being used. However, it helps in amplifying the signal. Then you have sandwich ELISA where your antigen is sandwiched between two antibodies. So first you coat the plate with antibody. So you remember sandwich ELISA is where you coat the plate with antibody instead of antigen. In all others you coat it with antigen. So you have your antibody specific to your antigen at epitope A, and then you have a detection. So this is your capture antibody because it is helping in capturing your antigen. And then you have a detection antibody which has epitope for which is against epitope B, and then this is a primary, and then you have a secondary for your primary, which is attached to an enzyme. Then you have your substrate, and enzyme substrate reaction occurs, and then you get your color. This is highly specific and it is very sensitive. Then you have competitive ELISA, where you have your antigen bound to the plate, and then you have your sample in which you have your antigen. So in your sample in which you have your antigen, you will also add your antibody. So your antibody antigen complex is present in the sample. But depending on the amount of antigen present, you might have free antibodies or you might not have free antibodies. So if the antigen titer is less, you will have freely available antibodies because it will be in excess now, and those freely available antibodies will bind to your antigen on the plate. So this means that the antigen was less. That is the reason we have more signal here. But if your sample has more antigens, all your antibodies are used to antigen antibody complex in your mixture itself. You don't have any free available antibody to bind onto your plate. So your color here will be less or it will be zero depending on the amount of antigen present. So if you have high antigen in your sample, here your color will be less, and if you have less antigen, your color will be more. Hope that is clear. So this is how different types of ELISA are performed. So to summarize, direct ELISA uses one antibody, indirect ELISA uses two antibodies, and competitive usually uses one. Sometimes it can also use more than one for amplifying. And then sandwich ELISA uses three antibodies: capture antibody, detection antibody, and enzyme linked secondary antibody. Then next is another question about blood grouping. So in a blood grouping test, if the sample blood agglutinates with anti-A antibody, then the blood group is, can you answer? Is it A, B, AB or O? So you have a blood which agglutinated with antibody A. So which blood group can it be? Anybody? Are you there? Yes, guys. Okay, just now we discussed. So if it is agglutinating against antibody A, that means it has antigen A. So antigen and antibody together will agglutinate. So this is how blood grouping is done. So you have sorry, you have your A antigen here, and you have your B blood group here. So I think I made a mistake. Sorry, I think sorry. The answer is B.
Answers B, I think. So.
A or B? One second. I'm getting confused. Yeah, it is A. Correct. So yeah, it has antigen A. So when you add anti-A antibody, it will agglutinate. Then if you have a blood which is B, if you add anti-B antibody, it will agglutinate with B. If you take the sample and add individually antibody A in one drop and antibody B in another drop, if it is an AB blood group, it will agglutinate in both. I think this picture is confusing. I better delete this. Yeah. So you can see here in this case, yeah. So when we do blood grouping, we add all the three A, B, and D. D is nothing but RH. So it will give you whether it is positive or negative, RH positive or RH negative. So in this case, one here, you can see the blood has agglutinated with antibody A and antibody D. But there is no agglutination in case of antibody B. So it is positive for both A and B. That means it is A positive. And then in this case, there is agglutination seen in case of B and D. So it is positive for antibodies both A and RH. So this is B positive, and there is no agglutination in case of anti-A antibody. Similarly, in case 3, we can see agglutination in all the three. We can see agglutination in A, we can see agglutination with antibody B, we can see agglutination with anti-T. So this is AB positive because it has all the antigens which are able to agglutinate with the antibody. In case of this, you can see uh, there is no agglutination with anti-A or anti-B, but there is agglutination with anti-T or RH. So that means it does not have A antigen, it does not have B antigen, so there is no agglutination, so it is O blood group. But however, it has agglutination with RH, that means it is RH positive. So this is O positive. So this is how blood grouping is done depending on the agglutination. If you have that particular antigen, when you add antibody against it, your blood will agglutinate. So it is positive for that particular antigen. So next is the individual with absence of anti-A and anti-B in their plasma belongs to which blood group? Can you answer this? So here they are talking about the person's blood itself, blood plasma. So the blood of that particular person does not have antibody A, does not have antibody B. Which blood group person will not produce antibodies against A and B?
Option D.
Yeah, it is option D, AB, because they already have those antigens, so it is self antigens for them. So this is another summary table. I think this will be helpful. Yeah. So here you have blood groups, different types: A, B, AB, A, O, all positive. Then you have A, B, AB negative, and O negative. So your antigens on this are A, antibody present in the which can be produced in the blood of this is B, and they cannot receive blood from A negative or O negative. Sorry, they can receive blood from A negative or O negative because they they cannot produce antibodies against A. And O already does not have any antigens, so that will also not stimulate the production of any antibodies. And since it is negative for RH, they can easily receive the blood. And people with blood group B positive or negative can receive blood from B negative and O negative because they have antigens B and produce antibody A. Similarly, people with AB, since they have both A and B, they do not produce any blood plasma, so they can receive the blood from all the groups B, AB, and O. So these are known as universal donors. So, however, only the AB positive here can receive the blood from all, but AB negative cannot receive from positive of RH positive or of any group. It can only uh, receive blood from negative. So you can take the blood from A, B, AB, O in case of AB. And in case of A positive and AB negative, antigen present on them are A and here in this case AB, but this is RH negative. So here you can get blood from A negative, B negative, AB, O negative. And in case of O positive and O neg, so here in O positive can receive only from O because if it receives blood from A, B or AB, all the antibodies will be produced in their blood except for RH. In case of O negative, this is very rare. So it can produce antibodies against all the three. A, B, and also against RH if received from any other blood group other than O negative. So O can, however, give blood to all the other blood groups. So they are known as universal donors. Okay, so this was about blood groups. Then the other question was regarding radial immunodiffusion, which is RID. So, radial immunodiffusion is a quantitative immunoassay used to measure specific antigen concentrations. So, based on antigen antibody precipitation in a gel, this works. So, your antibody is uniformly embedded in your agarose gel. So, you have your plate in which you have your agarose gel, and then your antibody is uniformly embedded in the gel, and then antigen is placed in wells cut into the gel. So you can see here, this is your plate, this is your gel, and then you cut out circular agarose uh, from the different places like this. So basically you puncture it with a cylindrical straw or anything, and then you remove the gel from that area. Then antigen is placed in these wells. So when you puncture and remove it, you basically make like a well in the agarose where you can now add your antigen. So antigen is placed into these wells, cut into the gel, and then your antigen which you have placed here will now slowly diffuse into your gel. So precipitation occurs wherever equivalence zone is reached. The equivalence zone is nothing but the concentration of the antigen equals to the concentration of antibody required to form a stable lattice. So we have something called as antigen antibody complex, right? In previous case also we saw where uh, agglutination happened, but here uh, a stable lattice will be formed it because you have cut it in a circular shape. The diffusion also will be circular. So radial means circularly outward equally. So the formation of a ring appears. So you can see here your antigen is more in this region and antibody is comparatively less. Here you can see at this particular ring region, the antigen antibody concentration is optimal for a stable lattice formation. So it will precipitate in that region. So stepwise events occur. Antigen diffuses outward from the well. Antigen meets antibody in the gel. And when the local concentration reaches the equivalence point, immune complexes form. So these complexes will now precipitate there, and you will see a formation of a ring-like structure. So forming a visible ring around the well. The diameter of the ring increases with antigen concentration. So if you have more antigen, it will need more antibody for that equivalence zone to form. So the diameter of the ring will increase if you have more antigen. So the standard curve in RID. So to quantify unknown antigen concentration, a standard curve is prepared. So how it is done is we prepare the wells of known concentration antigen, allow them to radially diffuse, then measure the diameter of these rings, precipitant rings, or after appropriate incubation time. Then we plot the diameter on the y-axis and antigen concentration, known antigen concentration. This is our standard on the x-axis. Then we will get a standard curve. Then the relationship is linear and early range, and at higher antigen concentration, the curve may deviate due to saturation. Then determining the unknown antigen. So when you already have a standard graph now, similar to protein estimation, you can place an unknown sample in the well, measure its precipitin ring now, and then compare the diameter of that to the standard graph. So you can determine the concentration of that antigen. There are types of radial immunodiffusion. One is the Mancini method and one is Octoluni method. So in Manzini method, it is simple, single diffusion in a gel with pre-embedded antibody. And in Octoluni method, it is double diffusion and it is mainly qualitative, used for antigen antibody identity test. If you want to identify a particular antibody, it mainly use Octoluni. Manzini can be used for quantitative measurement for the concentration of antigen. So you can see here, this is the standard graph that we plot usually of known antigen concentrations on the x-axis and precipitin ring diameter on the y-axis. So you can see it is linear. So as the antigen concentration increases, the precipitin ring diameter also increases. So this has a particular threshold after which it might reach saturation. So the linearity is taken only in that region. So more antigen, larger the diameter, ring diameter squared is often linearly proportional to the antigen concentration. Only antigen antibody equivalence zone produces visible precipitate. So, to summarize, standard curve in this is drawn between antigen concentration and the diameter of the precipitate ring. So this allows the quantitative determination of antigen in unknown samples using the standard curve. So that was it about the RID. Then we have something known as streptavidin. I think we already discussed about streptavidin in the previous sessions. So we'll quickly go through it again. So streptavidin is a tetrameric protein derived from bacterium Streptomyces avidini. Each monomer can bind to one biotin molecule. Streptavidin biotin are usually used as pairs in experiments for detection. So the tetramer binds to four biotin molecules. It has extremely high affinity for biotin. The KD is approximately 10 power minus 14. So lower the KD, higher is the affinity. So it is one of the strongest non-covalent interactions. And the source is from soil bacterium Streptomyces avidini, and it is tetrameric. It has a binding pocket for biotin, no glycosylation, unlike avidin. So it's more better than the avidin. Avidin is the main molecule, and streptavidin is the tetrameric protein. So, functional and application in experiments or in lab research is streptavidin binds to biotinylated molecules like proteins, DNA, antibodies, and it is commonly used in immunoassays, immunohistochemistry, so ELISA, Western blot, etc., where biotinylated antibody binds to antigen, and streptavidin conjugated to the enzyme or fluorophore allows detection. Then, in case of IHC and imaging, fluorescent streptavidin binds to biotin labeled probes for microscopy. Then, for purification, also it can be used where streptavidin can be coated to the beads and can pull down biotinylated proteins or nucleic acids. It is also used in DNA RNA detection by labeling, and detection is done uh, when streptavidin binds to the biotinylated nucleic acid. This allows visualization, quantification assays like dot blot, northern blot, or fluorescence in case of fluorescence in situ hybridization, which is FISH for RNA DNA detection. So this is a table of the source, glycosylation as electric point, binding, biotin binding affinity, and non-specific binding, etc., between streptavidin and avidin, and how streptavidin is better than avidin. Then why choose streptavidin? Because low non-specific binding, better for sensitive assays, more stable under a wide range of conditions. So this was about streptavidin. Now we have something about ELISA. So we discussed all four types of ELISA a while ago. So I think you can answer this question. So the process in which the target antigen is first coated onto the multivalent plate and then detected by an enzyme-linked primary antibody. So what type of ELISA is this?
Maybe direct ELISA.
Yeah, correct. So we have only one antibody which is coated to the enzyme, and then we have an antigen which is coated onto the plate. So it is direct ELISA. You have your plate, you have your antigen coated, you have primary antibody bound to enzyme. Then when you add a substrate, antigen antibody reaction will give the color. and this was other types of ELISAs which we discussed. So I think that is it. Do you want me to explain, re-explain any topic or any question? Let me know if there was any disturbance or if there was no audio during any time, or if you have any doubts in these questions. Anybody, any doubts?
No, ma'am. Thank you so much.
Okay, so I hope it was clear. So if there are no doubts, I think we can end the session now. Thank you for joining.
Thank you, ma'am.